Hello everyone, in this video we will give you an overall introduction of BrainCog. In addition to providing different basic components to facilitate the development of brain-inspired artificial intelligence and brain simulation, BrainCog also provides many basic examples for the researchers to learn and use. For the basic components of BrainCog, we have offered different neurons, the encoding strategy, the brain areas, and the different learning algorithms. So we provide different neuron types in a node file under the BrainCog base directory, such as the commonly used the if neurons, the leaf neurons, Ijkovich neurons, and the HH neurons. Users only need to inherit the base node and define their own memory integration function like this, and their own spiking fire function to define their own neurons. Also, we define different encoding mechanism in the encoder file under the BrainCog base directory. As can be seen here, we have offered the rate coding, the face coding, the temporal coding. Users can choose different encoding types according to their own needs. So when the researchers want to define their own algorithms, take the back-EI net as the example. First, we define the basic computing component as the back-EI node in the node file. It will integrates the self-feedback connections and the excitatory and inhibitory neuron balance. As can be seen here, when we define the self-feedback and the balance of the excitatory and inhibitory neurons, the whole information flow can be seen like this: the memory integration function, the spiking firing function, and we use the back-EI node as the basic computing component in the whole forward pathway in the net. And as can be seen here, we use the encoder to encode the inputs as we want, and we use the file rate as the final output. And when we run the main file, we can get the output, or we can get the results we want, as can be seen here.
BrainCog also integrates multiple learning rules, functional brain areas, and different neural network connection modes, such as lateral inhibition, dropout. In custom linear, you can design your own network architecture by entering the connection weight matrix and connection matrix of the network. In terms of learning rules, BrainCog integrates BCM, Herb, SCDP, STP, and other learning rules. By calling the above basic components, we can build multiple functional brain areas. Take the basal ganglia as an example. By calling the neurons, learning rules, connections in the base, we can build excitatory and inhibitory connections between different nuclei in the basal ganglia, and give the synaptic update rules to the connections. The main rule of the basal ganglia is to make decisions. So we design a brain-inspired decision-making spiking neural network in model zoo, with the basal ganglia as the core and other neuronal clusters to form a more complex brain-inspired decision-making spiking neural network model. The input of the model is the current state of the environment, and the output is the selected behavior. The brain-inspired reward-modulated synaptic plasticity is used to update the weights of the network. Inside the decision-making below the example, we use the game Flappy Bird for validation. By calling BDMSN inside model zoo, combined with the game of Flappy Bird, we can validate the model. The experimental results show that the bird is able to learn the correct strategy during interaction with the environment. In addition to decision-making, BrainCog covers multiple brain-inspired cognitive functions and brain structure and function simulations. The cognitive functions include decision-making, knowledge representation, and reasoning, such as sequence production, music memory, and stylistic composition, and motor control, perception, and learning, such as the multisensory integrated concept learning, quantum superposition inspired spiking neural networks, and supervised learning spiking neural networks. And the social cognition functions includes self-perception, theory of mind spiking neural networks. These cognitive functions are implemented based on the basic components of the BrainCog.
The following most brain simulation as an example to give us a brain introduction. Open the examples folder on the BrainCog main screen, followed by the multi-scale brain structure simulation subfolder. And we can see those folders. And we open the mouse brain folder. According to README, download the weight metric of 213 mouse brain region connections published by the Allen Brain Institute from the web link and place it in the field for background running. Download the Python file from mouse model to the same folder. The ratio of different types of neurons and the parameters of neurons and the time of simulation can be set according to the requirements. And we copy the Python file to the local document. And taking the 200 millisecond simulated mouse brain model as an example, a MAT date will be generated after local running. And the drawing software of the program will automatically present the scatter diagram of the whole spiking brain. And this is our mouse brain model. Thank you very much.